Yeah, first of all, thank you for the kind introduction. And today I'm going to talk about adaptive sampling, which is the nanopore sequencing technology, and how you can use it in clinical metagenomic sequencing. So I separated my talk into subtopics, which is first the nanopore sequencing, how it works, what is adaptive sampling, and then we take a closer look to its clinical applications where you can use adaptive sampling. So nanopore sequencing is a long-read sequencing technology where you can sequence very short fragments, for example, but also very long DNA molecules up to several megabases. It's a fast and real-time sequencing technology. That means basically, as soon as you start the sequencing, you get data from it and you can directly analyze the data. It's a single molecular sequencing technology. That means every DNA strand that is passing the nanopore is generating its own electronical signal, which is a change in ion current. And this then can be translated to the DNA bases. We also have the opportunity to detect base modifications with nanopore sequencing, for example, methylation patterns. And in 2020, Oxford Nanopore introduced a new method for nanopore sequencing, which is called adaptive sampling. So you can basically translate it to targeted sequencing. So what is adaptive sampling? So adaptive sampling gives us the opportunity to reject or accept sequences during sequencing. For example, a DNA strand is being sequenced, and if it's not of interest for us, then the DNA strand will be ejected out of the nanopore, and then a new strand attaches and will be sequenced. So it all happens on a strand-by-strand basis, and it means for every DNA molecule, it will be checked against a reference sequence and will be then ejected or continued to be sequenced. This gives us then the opportunity to do target enrichment. For example, if you only want to sequence antimicrobial resistance genes, you can enrich only them. Or for example, if you have samples with a contamination of, for example, human DNA, can do human host depletion and only sequence microbial DNA. Cool thing is also that you don't need any additional sample prep. You just switch a button on in the sequencing software and then adaptive sampling is enabled. Okay, so what are the requirements for adaptive sampling? First, you need to decide the mode of action. That depends actually only on your experiment and what you want to show. Do you want to do target enrichment or target depletion? Then you need a reference sequence you want to enrich for or deplete. In my case, it was the human reference genome, which I wanted to deplete. And third, I chose a fast base calling model, which you can also choose in the software. It's very simple. Just click, select the model, and then you can start the sequencing. And basically what happens in the sequencer is what I explained in the slide before. DNA molecule attaches to the nanopore, then it will be checked, is it a sequence of interest? If yes, then it will continue sequencing. Or if it's a sequence that is not of interest, then it will be stopped. The sequence will be, or the fragment will be ejected and a new DNA molecule will be started to be sequenced. Okay, now you know the basics about adaptive sampling. Now we have a closer look how it performs in metagenomic sequencing and in clinical applications. So first, a little clinical background. So we are working closely together with the obstetrics department of our hospital, and they have pregnant women sometimes which suffer from bacterial vaginosis. Bacterial vaginosis is the disruption of the composition of the vaginal microbiome, which can lead to pregnancy loss or preterm birth. So it's a really relevant topic. And current identification of bacterial vaginosis is culture-based diagnostic. And it suffers basically from long reporting delays and low sensitivity and specificity. And then the obstetrics department approached us and asked, could we do something with nanopore sequencing? And I tried on some samples and sequenced them, and I found that they have really high amounts of human DNA, so around 75 and up to 98% of human DNA. And the solution for us was then, okay, let's try adaptive sampling on real clinical samples, because until last year it was only shown for mock communities, so artificial communities that were sequenced. And yeah, let's have a look at how it performs. So first we had to decide which mode of action do we want to do. Do we want to do the depletion or the enrichment mode for metagenomic samples? First, we did a control experiment. So without adaptive sampling or any special sample preparation. On the y-axis, you see the relative abundance of some organisms, Gardnerella, human lactobacillus, and other bacteria. And the control basically gives us the ground truth of the experiment. So the sample contains here of around 75% human DNA. Then we did the same experiment with the same DNA, but we now used adaptive sampling with depletion. So we used human host depletion, and you see that the fraction of human reads that were sequenced is significantly lower. So we have now 35% human DNA being sequenced. And then we did the same experiment again, but with adaptive sampling with enrichment enabled. We enriched for the 10 most abundant organisms in the control experiment, and we found that we have only 7% of human DNA being sequenced. So now you would guess, okay, best for human DNA depletion is the enrichment, but we further analyzed our data and we counted the microbial bases, and we found that enrichment yields only 0.77 gigabases of microbial bases, which is very few in comparison to the depletion experiment and the control experiment. So also from a logical standpoint, if you get an unknown sample, you don't know what to enrich, but you know that there's human contamination or human DNA contamination. So the logical thing is you go for human DNA depletion. And that's also what we did for the following experiments. So we now assess the performance of the human host depletion. Therefore, we got 15 vaginal samples. They were sequenced first without adaptive sampling enabled, which is the control here. And then we sequenced the same samples again, but with human depletion enabled. So as you can see, we got around 1.7 times more reads in the depletion experiments compared to the control experiments. This translates also roughly up to, if you want to generate the same amount of data using no adaptive sampling, we would have at least need 1.7 flow cells. So basically 2 flow cells for sequencing. So next, we were interested in where can you find the bacterial DNA and human DNA. Here again, we have the control experiment and the depletion experiment. On UIX, we have the relative abundance of bacteria DNA and human DNA. And you see that the fraction of human is really high in both control and depletion, and bacteria is low in control and depletion. So they are very similar. But when you do adaptive sampling, the sequencer produces basically 3 different read fractions, which is the accepted read fraction, where you basically have only microbial DNA, as you can see here, but also a few human DNA reads. Then we have the no decision read fraction and the rejected read fraction. The rejected read fraction is very specific for depletion, so we have around roughly 99.98% of human DNA in this read fraction, and the rest is, not significant bacterial DNA. But important from this slide is you have the no decision read fraction, which still contains bacterial DNA and human DNA. So human depletion is not completely possible with adaptive sampling. So when we do metagenomics or metagenomic sequencing, we want to represent the composition of the microbial sample as close to the real life as possible. And that's why we compared the genus of the samples in our control experiments, which you can see here in gray, and in the depletion experiments, which you see here in blue. And the takeaway here is basically that both experiments show the same abundance of genus or organisms in all around the samples. So adaptive sampling is not altering the microbial composition, which is very important for metagenomic or clinical samples. So the takeaway from my talk is basically you can do adaptive sampling additionally to your wet lab depletion protocols. With adaptive sampling, you get the depth of 1.7 flow cells, which basically means you can put more samples on a flow cell and get the same amount of data, which then reduces the cost per sample, which is very important also for the hospital. And microbial composition is not changed, which is actually pretty nice. And the only negative point here is that human DNA could not be completely removed from the sequencing experiments. So keep that in mind when you do adaptive sampling. If you want to know more about this adaptive sampling topic, you can check out our paper, or you can text me on Twitter or write me an email. And yeah, thank you for the opportunity to have a talk here, and enjoy the next talk.